All right, happy spring. Hopefully will snow less as the day goes on, right? I wanted to remind you, a week from today, final exam. I had 2 students last year that came at normal class time at 1:30. Everybody else was pretty much done. So make sure you come at noon next Monday and not at 1:30. All right, a week from today. And I also wanted to talk just about 2 questions, 2 of the most commonly missed questions from our last exam. This question really was asking, I could have asked, okay, what are the stages of phagocytosis? So if you think about the stages of phagocytosis, you've got the phagocyte chemotaxing toward, following cytokine signals to where the site of infection is. We talked about how it recognizes that it's appropriate to phagocytize that pathogen, so the toll-like receptors, and then how it has to adhere to the pathogen. It engulfs that pathogen in an endosome, and then inside of the white blood cell, it has to fuse with a lysosome. That endosome fuses with a lysosome, and that's when all the digestive enzymes are, put on that pathogen. Then it usually kills it, and then the phagocyte eliminates it. So basically, if you've got a pathogen that can prevent the formation of phagolysosomes, that means it's preventing the lysosome from fusing with the phagosome. So essentially, that just means then that the pathogen just hangs out in that endosome inside the white blood cell, and it doesn't get digested. All right, so that was correct answer there. The other commonly missed one, I think, that T-dependent antigen and that T-independent antigen, like I still have to think through that every time to think about what the difference is between those. But we talked about this, and I think maybe I wrote it on that slide when we were talking about T-independent antigens. Remember, those are antigens that don't need help from T helper cells, that they will stimulate our B cells on their own to start differentiating into plasma cells and make antibodies. So not all, there's just certain bacteria that have capsules that can serve as T-independent antigens. So that was correct answer there. Okay, so still time if you want to come to my office at some point this week and look over that last exam, look over things that you've missed. Still time to do that. I will be around Friday, for sure in the morning, and I can stay until about 2 o'clock if I need to. So let me know if that is a time that works for you, that you want to go over anything. Okay. So where we left off last time, we were talking about gonorrhea and talked about some of the virulence factors. The big thing about Neisseria gonorrhea, remember, that's caused by a gram-negative pathogen, a gram-negative cocci. Okay, so a round cell. The big thing that pathogen can do is the antigenic variation and the phase variation. So remember, it has this really wily way of changing the outside, the outside of its cell that's the most antigenic thing to our immune system. It changes the proteins that make up the pili. So it stays ahead of our immune system, and we make antibodies, and they don't do any good because the pathogen has changed already. So, and then the symptoms are quite different between males and females. males usually have a lot of pain and get treatment right away, where in females, it's often asymptomatic. So that doesn't necessarily mean that it's not a problem. It can potentially still, be multiplying and cause things like pelvic inflammatory disease, which can lead to infertility. It can lead to infertility in males too, but they usually get treated, and that doesn't usually get that far. Okay, so that's kind of where we ended last time with the symptoms. In terms of incidence is rising. We talked about how this is, I think we mentioned, it's the second most common sexually transmitted disease, or sexually transmitted infection, sometimes we say, behind chlamydia. But oftentimes people will have chlamydia and gonorrhea actually transmitted at the same time. So the numbers I have, as of 2018, around somewhere on the order of 580,000 cases in the US. This is one of those diseases that's notifiable. So you get diagnosed at the doctor and they notify the CDC. Probably we're way undercounting this, again, because so many women don't realize, maybe even for a long time, that they don't have it. And interestingly, the cases here, so there's a couple different ways that we can express how many people have this infection or any infection. We can say the total number of cases, or this figure in your book, you can see that it shows incidence per 100,000. So remember the difference between those two terms, incidence and prevalence. Incidence is the new cases, and then prevalence is just how many people already have the infection. So if you express incidence per 100,000, then, over time, as our population increases, right, you still have kind of a good gauge of, even though population has increased, what the numbers that we have. So if you look at the total number of cases in the US as of 2020, actually, I have as of 2019. Okay. So I should have updated this. We have about 600,000 cases as of 2019. Oops. And that represents almost a doubling of cases since 2009. So we had about 300,000 in 2009, almost double that much only 10 years later. And again, if you express that per 100,000, you can see we're having an uptick here. Not as many as what we probably had, early 80s before the AIDS pandemic. With that, people were afraid of getting AIDS, so they, had safer sex or less sexual partners, and we saw the incidence coming down. It's kind of going in the other direction again. Of course, one of the reasons that I bring this up is that you all are in the most common age group. Of course, the main risk factor is having, the more sexual partners you have, obviously, the more likely you are to contract it. And you can get it over and over again, right? Because it changes so much, you don't have any memory cells that will work for a new infection. Okay. So again, it's out there, not just sexual partners, but of course, IV drug use is another risk factor. Sharing dirty needles is another way. It's not just sexually transmitted, remember. So what about treatment? Right now, it's really very interesting. This YouTube video is, they just updated it, made by the CDC. It's just like a two-minute video that talks about how we've changed treating gonorrhea over the years, and how we've had to, we use this antibiotic, and everything gets resistant to it. We've had to switch to do this, and then this wasn't useful anymore. So it's kind of a interesting look. It helps you, realize the problem that we have with resistance. Right now, we're using a cephalosporin, ceftriaxone. It's injectable. Up until a few years ago, they were recommending that you use that drug in combination with azithromycin to try to slow down resistance, but they were starting to see some resistance to azithromycin, and so they recommended stop using that to treat this because they want to save erythromycin for some of those, childhood illnesses that it's still very effective for, like strep throat and ear infections and that sort of thing. So that's the new recommendation. There are areas of the world, particularly Southeast Asia, where they're seeing strains that are resistant to all the antibiotics that we have. And so they're kind of areas of the world. There are areas of the world where they're really pushing, public health campaign, abstinence, monogamy, condom use, because again, they're really worried that we're close to not being able to treat this very effectively. There is no vaccine. There are some that are kind of in the works, but right now it's not a priority because we still do have antibiotics that treat it pretty well. So if that should ever not be the case, then I'm sure that we will eventually come up with a vaccine that is, at least somewhat effective, but it's not, I think there were, I don't know if it got to the point of clinical trials yet or not, but for now, there isn't. No. Okay, any questions about gonorrhea? All right, so a related organism, another Neisseria, we're going to talk a little bit before we get into some viral infections today. Neisseria meningitidis, okay, also, again, gram-negative cocci often presents as diplococci, pairs of cocci together. Like Neisseria gonorrhoeae, this is a human infection, so we're just passing it around, obviously, between humans. And in fact, for Neisseria meningitidis, about 10 to 40% of us are considered carriers. Okay, usually in the upper respiratory tract. Okay, and that's really quite interesting because, as we'll see, this is a pretty heinous infection, but some of us, have it in low numbers in, our nose, upper respiratory tract, maybe just transiently for a little while, and then it's kind of outcompeted and we lose it again. But we are, again, just humans passing it around to other humans. It's droplet spread. So remember, that's close contact, closer than a meter away. Usually things like sneezing, coughing, or some kind of fomite transmission. Remember what a fomite is? That's some non-living object, like a drinking glass, for example, or maybe a fork, something like that, a eating utensil. Okay, share it with somebody else that's either maybe is a carrier, and then you, I don't know, get it in larger numbers. Okay. So the disease that we're going to talk about is meningococcal meningitis. So we talked about other bacteria that can cause meningitis, like Staph aureus or Streptococcus pneumoniae. And of course, meningitis is inflammation of the meninges. It's a kind of three-part membrane that surrounds your brain and spinal cord. Not many organisms that can pass that blood-brain barrier, but again, there are a few. Haemophilus influenza is another bacteria that can cause meningitis. Meningococcal meningitis is a little bit different because there's a little bit different toxins involved, and it's primarily endotoxin. So we'll get to that. Of course, there are some viruses that can cause meningitis. Viral meningitis is not nearly as severe, which is usually we don't have as good treatments for viruses, but you don't get the toxin production, the exotoxin production or endotoxin production from viruses, of course. Okay, so what do the early symptoms of meningococcal meningitis look like? Thankfully, this infection is really rare and actually is kind of getting more rare. That's the trend. So that's good. So early symptoms, fever, sore throat, nausea. So it kind of starts out kind of flu-ish type symptoms, maybe. Of course, you don't usually get nausea from respiratory influenza. But the other symptoms, stiff neck and headache. Those are the real red flags there. Not just a headache, but like the worst headache you've ever had in your life. Like really, you know, you can have a stiff neck, you sleep wrong or whatever, but just, yeah, that's going to be accompanied by, again, the worst headache you've ever had. And if you or somebody that you know has those symptoms, you should go to the ER because this is an infection that can move very quickly. Within a number of hours, that can move into some of the later symptoms that are more serious here. And the reason that it progresses so quickly, that's because when the organism is replicating or making more, reproducing itself, it's actually releasing its outer membrane. Okay, remember what's part of the gram-negative outer membrane? Lipid A is part of the lipopolysaccharide, LPS, and another name for that lipid A is endotoxin. And this organism, usually when a gram-negative, the only time a gram-negative releases endotoxin is maybe to some small extent when the cell's dividing, more so when the cell dies and the cell, that outer envelope just kind of starts to break up and disperse. But Neisseria meningitidis has a way to slough off that endotoxin all the time. And we call that blebbing. Okay, so it's not releasing endotoxin just when it dies, but when it's alive. And that endotoxin is somewhere from 100 to 1,000 times more toxic than I would say kind of your normal, you know, gram-negative lipid A endotoxin. Okay, so it's nasty stuff. It's releasing it in large quantities. So that's going to cause some of the lost my train of thought here. Some of these later side effects. Okay. So if you remember this little here, if you remember this little graphic that we talked about last time, the effects of lipid A, things like inflammation. If you get enough inflammation, right, patient's blood pressure can go down, they can go into shock. We've got this blood clotting reactions that are inappropriate that can be caused by that endotoxin. So we start getting that inappropriate blood clotting, which can cause these black spots. It starts out as kind of a red rash, and then it just turns kind of more purple due to the capillaries breaking, and eventually necrotic. Okay, these black spots that we call petechiae. Okay, so due to that endotoxin release, breaking capillaries, and eventually the petechiae will fuse together to form like black areas, especially on the limbs. Okay, so that's areas of necrosis. That looks like a baby will probably have to have that hand amputated. Once it's black like that, it's dead. Patients can go into shock because their blood pressure gets too low from that, again, that dilation of their blood vessels. A really high fever can cause neurological effects like delirium. We go into cardiac arrest. Without treatment, with these later signs and symptoms, then patients usually die, almost 100%. With treatment, the prognosis is a little bit better, maybe have like a 20% mortality. but of those that live, maybe 10 to 20% have some disability. So either, so 20% of survivors. So things like loss of limb or mental retardation, blindness, deafness, like serious forever chronic issues. So again, really serious disease, something that you don't want to mess around with. How do they diagnose? They will do a lumbar puncture, and that's how they can tell for sure if you've got, you know, the bacteria in your spinal fluid. Probably would start you on antibiotics before they mess around with doing that if you have all the signs of symptoms of that. Okay, so pretty serious infection. Who's most at risk? This is one of those T-independent antigens, the capsule. We said Neisseria gonorrhea. I didn't make a capsule, but Neisseria meningitidis does make a capsule. And it's a T-independent antigen. So remember, really young children don't have as good a response to that. Their B cells are just less mature. So when very young children are exposed, they are more at risk. And then the second most at-risk age group is young adults. So teenagers, college students. Why is that? That is, I haven't really read anything about that, but usually the outbreaks have stemmed from some party somewhere. So, you know, passing drinking glasses around, things like that. It's a rare infection. So if you get someone that actually gets it twice, they start looking to see if there's any immune irregularities. And often they find that people that have a complement deficiency are going to be more at risk. They may not even know they're complement deficient until maybe they get this infection again once or twice. But anybody that's healthy can come down with this. It's not just an opportunistic pathogen. If you don't have individuals that don't have spleens are more at risk for bacterial infections in general, and of course anybody that's immunocompromised. That's just kind of a catch-all. Okay, and I just thought I'd show you these kind of recent statistics. I just got these off the CDC webpage. Like I said, it's not a very common infection. Most at risk here, you know, young children. There's a blip here, young adulthood, and then just as we age, our immunity wanes quite a bit. And I thought this was interesting. A look at the incidence of meningococcal meningitis incidence since about 1970. So you can see today we're really low. We do our first vaccine to this came out in 1978. So, like right here when it was at its height, we came out with a vaccine, and that probably had something to do with the incidence getting lower. We came out with the vaccine that we use now in 2005. So, it was well kind of on its way down, but, presumably was better than that first vaccine even. So, so again, it's pretty rare. Let's look at the virulence factors, and then we'll talk a little bit more about that vaccine and what we do to treat it just a little bit. But some of the virulence factors are pretty much the same as those that we talked about with Neisseria gonorrhea. And I realized right before lecture that I spelled that organism wrong. You throw that O in there, and you don't really hear it. So anyway, I did spell that wrong. But so remember, the Neisseria gonorrhea has the pili. The book called it fimbriae. Most sources call them pili, but therefore attachment. Of course, the Neisseria gonorrhea is attaching to different tissues to the urogenital tract, and Neisseria meningitidis is attaching to the meninges. It has protein adhesions specific for the meninges. Oh, and also endotoxin. We mentioned that already, how it's shed. We call that glebbing when it's shed in large amounts. And again, it's just a little different variety that's even more toxic. Unlike Neisseria gonorrhoeae, it does make a capsule, and that, of course, is going to help protect it from phagocytosis. But it actually is kind of a good thing in some ways that it does make a capsule, because we actually have our vaccine that's effective against parts of those capsules. So there are many different variations of that capsule. So we kind of split that organism into different serogroups or variations of that capsule. Again, let me just, of the capsule. And the main ones, 95% of meningitis are due to the 5 of those serotypes, A, B, C, Y, and W122. Whoops, sorry, 135. Oh, a little bit crazy naming that last serotype. Okay, so A, B, C, Y, and sometimes they just call it W, or sometimes you see it called W135. Okay, so again, like 90% of disease is caused by those serotypes. There are other ones. Okay, so treatments, antibiotics. I'm pretty surprised that penicillin would be used to treat anything in this day and age, but I've read that a few different places. Cephalosporin, of course, one that passes, can chase that organism across that blood-brain barrier, probably one of the newer cephalosporins, later generations. Some anti-inflammatories to counteract that endotoxin release. It's such a serious infection that if you are exposed or have been around anyone that comes down with it, then you would be prescribed antibiotics prophylactically. So even though you aren't showing any symptoms, they will do that. I remember one of my first years of teaching here, we had a student that came down with this, and they vaccinated anybody on campus that wanted to take part in the vaccine, that wanted it, just kind of, again, because it is such a serious infection. So what about the vaccine? There are several different forms available. Again, the one that was approved in 2005, we call it MCV4. Okay, so that stands for meningococcal. So MC, meningococcal quadrivalent vaccine. Actually, I'm not sure if the V stands for vaccine or if the 4 is just quadrivalent, V for valent. I've also seen this called, when I looked on the CDC page a few days ago, they called it ACWY. Okay, and of course that stands for the different types of the capsule that the vaccine will cover. Okay, so quadrivalent, there's 4 different. So it's a subunit vaccine. It doesn't protect against that B serotype, because that apparently the sugars in that B serotype were just not as antigenic, so it was hard to get a vaccine that would protect against that. There is now a meningococcal serotype B vaccine, but you have to get that separate. I've seen ads for that on social media. It's so rare that's why it's just not commonly offered, but if you went to the doctor and said that you wanted to be vaccinated against it, you certainly could. It's a subunit. Both of these they're subunit vaccines. Okay, so they've taken part of that polysaccharide capsule and they have attached it to protein to make it more immunogenic. Okay, so we'll say for improved antigenicity. And I tried to look up the specifics about what protein that they used, and I think it's actually part of like inside the organism. Okay, but you could consider it a subunit vaccine. You could also consider it a conjugate vaccine because they're, again, putting lots of things together. It's pretty effective, somewhere between 85 and 100% effective. They don't give it to young children because it doesn't do any good. They don't make up a good memory response to it, so we have to wait till kids are around the age of 11 or 12, and then to get, you know, fully protected, they want to get a booster somewhere like 16 or maybe before they leave home, ideally, for good protection. Okay. The most, so like I said, there haven't really been any big outbreaks since, I think the most recent one was I want to say 2009 at Princeton, when 9 people were sick. I don't think anybody died from that one. And that was actually the B serotype, which up until that time had not been very common in the US. So that was kind of surprising, but again, fairly rare. Okay, any questions about either of the Neisseria? Okay, talked about that. All right, let's move on then to thinking about some viral pathogens. Okay. Yeah, so that's a good question. It's easy to mix those up. So antigenic variation, that's, we talked about, we're going to talk about how flu virus can do antigenic variation as well. But that's just something that the Neisseria gonorrhea is really good at, switching up the proteins on its pili. The phase variation is when it actually will detach the pili. So it's kind of more like an on-off switch. Either it expresses the pili or they're allowed to just fall off. So that's the difference between antigenic variation and phase variation. So yeah, good question. All right, so today we're going to hopefully get through some DNA virus pathogens. And that should leave us time for the RNA pathogens on Wednesday. Just a little review, like we've talked about viruses quite a bit as we've gone through our immunology chapters, but remember, it's important for the final that you think about what are the different kinds of microbes that we've studied this semester, and what's the difference between the different microbes? What's the difference between an archaea and a fungi and a virus and a bacteria? So think about some of those things as we think about the exam next week. Remember, what's special about a virus is that they can only reproduce when they're inside a living cell. Okay, that's so, do we consider them living? No, probably not. What are they made of? They are just nucleic acid. And remember, that's what's different about a prion. Prions don't even have nucleic acids, so they're really bizarre. Either DNA or RNA. They're not going to ever have both. DNA or RNA, and that's going to be enclosed in a protein capsid. Okay, so I just included that graphic, just different sizes and shapes of viruses. We had a couple different classification systems. We had the Baltimore classification that was based on what kind of nucleic acid they had. I don't have it memorized which viruses are in which Baltimore class for sure. We have the other hierarchical classification where we put viruses into like families and genus and classes. Okay, and then of course some viruses are enveloped, so they have this phospholipid membrane around them. Not very, I think there's maybe one bacteriophage that actually has an envelope, but it's mostly animal viruses, ones that infect humans, for instance. A lot of them are envelopes, not all of them, but that's much more common for that to be the case. All right, so in chapter 24, that's a chapter with DNA viruses. The only one that we're going to pick out and talk about is the family Herpes Viridae, because there's there's several different herpes virus that we're going to focus on. There are others in that chapter that we could talk about, like papillomavirus. That's the virus that causes warts and cervical cancer. In general, there's a lot more RNA viruses though than DNA viruses. Okay, so Herpes viridae, like I said, it's a big family of viruses, but they all have a few things in common. Okay, they are enveloped, as you can see from the little graphic there to the right. That's okay. They have a icosahedral capsid. So if you can remember, we had different capsids that we talked about: helical capsids, polyhedral. I think all the polyhedral ones are actually icosahedral, meaning it's 20-sided equilateral triangles there. So you can see this capsid over there. And it has linear double-stranded DNA for its genome. Okay, so you can see the long piece of DNA there. It's all one strand. If it was segmented, we would have different pieces of DNA there. Okay, and this is one of the larger genomes of any known virus. It encodes for about 80 genes. Okay, in general, the DNA viruses do encode for more genes. Some of the RNA ones, you know, maybe only have 5, 6, 7 genes that they encode for. So what's kind of unusual about the virion here is that they have a tegument, and it's not only Herpes viridae that have this, but it's very pronounced in Herpes viridae, and that's in the purple over in the graphic here, showing tegument proteins. So these are just kind of a collection of proteins that are carried in the virion, so kind of outside the capsid but inside the membrane, and these are just going to encode for proteins that's going to help it survive and start an infection. So there may be proteins that suppress the host immunity. For instance, there are proteins that will suppress interferon production in our cells. So remember, interferon was that cytokine that kind of helped to slow down viral replication. Some of our immune cells produce, or other cells as well. It might encode for proteins that kind of keep our cells from expressing MHC class 1. So remember, those are the glycoproteins that just kind of express what's going on inside of the cell. And if it's not advertising that, then our adaptive immunity is not going to be able to realize that that cell is infected with a virus. And then there's some spikes. We're not going to talk particularly about the spikes or name them for this virus, but help them bind to the correct kind of host cell and fuse with that cytoplasmic membrane. Okay. So still thinking sort of in general about all the herpes viruses, it is one of the viruses that's the most widespread. It's estimated that close to 100% of us have been infected with some kind of herpes virus, most commonly the kind that causes cold sores. Okay, so reservoir humans, it's true that animals have herpes viruses, but we think that they rarely cross species. There's one exception. I think there's a herpes virus that apes can get that they can give it to humans, but Again, the animal ones kind of stay in the animals, and humans, we pass it around to humans. So not typically zoonotic. All members of this family show latency. So remember, that's the lysogenic replication. That's what we call it in humans. So bacteriophage, we call that lysogeny. And that just means that the viral genome can integrate into our genome. And they actually don't incorporate right into the chromosome. If here's one of our cells, here's the nucleus, and here's our chromosomes in the nucleus. They don't incorporate into that chromosome. I should have drawn the nucleus a little bit bigger. They actually incorporate as episomes. Okay, so this is kind of, we describe it as a plasmid-like viral DNA that basically hangs out with the host cell chromosomes and gets replicated every time the host chromosomes get replicated, but it does actually not integrate right into our chromosomes. Okay. And you'll see that if you watch this little 2-minute video. It's a really nice video that just kind of walks through herpes virus infection into the cell. It's an animation. And because I don't know how to get the sound on in here, it's not easy for me to show it in class, but it kind of helps you visualize what's going on there. And the important thing to understand, even though they're not integrating into our chromosomes, once they're there, it's permanent. Okay, so the crux of that is, once we have a herpes virus infection, and unless our immune system fights it off from the get-go, they are going to be permanent in our cells, and we don't have a cure for that. Okay, so we might have some medications that help a little bit, but we don't have a cure. All right, and some herpes virus can be oncogenic. Okay, so there are a few of them that have the possibility of causing cancer. One is pretty rare. Well, both of them are pretty rare, but one causes a Kaposi sarcoma that we won't really talk about. That's usually AIDS patients that get that. And another one is I think it's a B cell. It's a B cell cancer. Burkitt's lymphoma. That's what it's called. Okay, so a few of them, depending on where they go latent and what kind of cells, they can be oncogenic. All right, so thinking about kind of how the virus infects our cells and the effects that it causes on our cells, they target mucosal membranes. So any little tear that you have in that mucosal membrane, think about around your lips, how in the winter when they're dry and cracked and it's very easy, you have little tears. That's where the virus can enter. Same thing with the urogenital tract. It's easy to have some little cracks in that. So they penetrate, the virions penetrate through the mucosal membranes, and they're going to multiply in those cells that are line the surfaces of those mucosal membranes, the epithelial cells, and then the basal cells right below that. They cause inflammation and death of those targeted cells. And when we're diagnosing somebody with a herpes virus infection, you don't typically take a body sample and look at it under an electron microscope. That's a really expensive, time-consuming way to try to diagnose a viral infection. Usually we look for some cytopathic effect, okay? And you see this abbreviated CPE. And this just means we look for a change in host cells due to viral infection, visible under light microscopy. So visible with light microscopy. Okay, so the cytopathic effect that we look for with herpes virus infection is syncytia. Okay, and that just means multi-nucleated cells. So you can see in both of these pictures, you can see the syncytia. So here's a nerve cell, all these little purple dots, those are all nuclei in that nerve cell. So a multi-nucleated nerve cell. And over here, this cell here has many nuclei in it. So if you think about it, this is a pretty sneaky way for herpes virus to infect other cells. Because if they're going to lyse a cell and break out of that cell, when they're outside of the cell, now all of a sudden they're more susceptible to our antibodies attaching to them. So if they can just get one infected cell to fuse with another cell that's not infected, okay, that's how they get multinucleate, when they're just fusing with other cells around them. But then they kind of outwit our, our macrophages and antibodies and things like that that might be more susceptible to if they're outside the cell. Okay. All right. So the different kinds of human herpes virus. And by the way, up until not that long ago, they were correctly called the herpes simplex virus. I don't know, for whatever reason, they call it human herpes virus. So HHV is more correct. You still see the HSV a lot of places too. But there are actually 8 different human herpes virus. I decided really I want to focus on these first 3. I'm just going to mention 4 briefly. But HHV1 is cold sores, sometimes referred to as above the waist herpes. And then HHV2 is genital herpes, sometimes referred to as below the waist herpes. So can you get HHV2 causing cold sores or the other way around? Yes, but generally that's due to oral sex practices. Okay. HHV3 is varicella virus, causative agent of chickenpox. So we'll talk more about that. HHV4, I just wanted to quickly mention that because some of you have experienced that. Lymphocryptovirus, also called Epstein-Barr virus, also the causative agent of mononucleosis. Okay, so which is essentially that herpes virus goes latent in B cells. So sometimes we describe mono as kind of our T cells and our B cells going to war against each other. So that's how you get that incredible fatigue from that infection. And there are some other ones that cause like asymptomatic infections, or like I said, like Kaposi's sarcoma is the HHV8. But we won't worry about those. We'll just worry about these. Okay. So a little bit more about HHV1, gingivostomatitis. That's the fancy name for fever blisters, sometimes referred to as cold sores. They are transmitted by close contact. Okay, so really close, like kissing or sharing a drinking glass, for example, sharing a utensil. Usually those sores last about 7 to 10 days. Oftentimes you have kind of a prodromal period where you might feel some tingling before the cold sore erupts. And then other sites where either HHV1 or HHV2 can manifest themselves. Sometimes you get sores on the fingers. We see that here. That's also called Whitlow's disease. That's the reason that dental hygienists wear gloves. So again, you've got lots of little cuts in your cuticle around your nail oftentimes, and if you're reaching in somebody's mouth that you know, has a cold sore, whatever, they can transmit that. HHV1 or HV2 can manifest in the eyes as well. We sometimes call that herpetic keratitis. And that can cause people to be really super sensitive to the light. In rare cases, can lead to blindness. And very rarely, the herpes virus can infect the brain or the spinal cord, causing encephalitis, which is an inflammation of the brain. That's obviously extremely serious. It's extremely rare. Usually only severely immunocompromised individuals, like patients that have AIDS. So untreated HIV. All right, I once again spared you the graphic pictures like I did with gonorrhea, but genital herpes targets the mucosal membrane of the urogenital tract. It's estimated about 1 out of 6 adults are infected with HHV2. So it's not the most common sexually transmitted disease. So chlamydia and gonorrhea are even more common than that. But of course, with herpes, once you have it, you don't have a way to get rid of it. It can get better over time, but you won't totally get rid of it. Typically, infected patients might have 2 to 3 outbreaks a year. Sometimes more frequently than that, sometimes every 2 to 3 weeks. So there's a lot of variability there. A sexual partner can pass it to another sexual partner, even if they're not having active lesions at the time. Much more likely to pass it on if they are having active lesions, but they can even without that. And it causes itching, some discharge, and sometimes fever. Okay. And of course, mothers can pass herpes on to their babies. Question?
Yeah, it's on the last slide. I'll look at it.
Oh, okay.
It was just about size. I didn't know if that's.
Oh, like the eyes?
Yeah, I don't know if that was like folliculitis or.
Yeah, I mean, I don't know a lot about that. Just that the herpes can, affect the, infect you through the eyes and cause little sores there around the eye. I don't think that's terribly common. Okay. Of course, mothers can transmit herpes during the birthing process. So obviously babies, they're going to take babies by C-section. If the mother's having an outbreak, maybe they would do it by C-section anyway, if they're aware that the mother sometimes has herpes outbreaks. Most commonly, it will pass on an eye infection to the newborn that kind of resembles the eye infection that we talked about with gonorrhea, where it can lead to blindness, but can move the central nervous system, can be much more serious, can be deadly, especially in newborns. So again, they're going to take babies by C-section. Whether you're talking about HHV1 or HHV2, remember they're latent. So basically, they are going to go latent in nerve cells. They can kind of hide out from our immune system in nerve cells, apparently. And they're just going to hide out in whatever ganglia sort of that they're closest to. So for HHV1, in one of the branches here of cranial nerve number 5, okay, the trigeminal nerve. So whichever branch they kind of move up to, that's where they're usually going to be latent. Whitlow, fingertips here, that's more likely going to be latent in the brachial ganglia, and then HHV2, genital herpes more likely in the sacral ganglia. Okay, and again, just from time to time, those they'll reactivate, start reproducing again, and travel back out to the ganglia to the site where the sores manifest. So what causes reactivation? Sometimes we don't really know. Sometimes it just reactivates, but most often there's some stress that will cause it to reactivate. So it might be something like a big dose of sunlight or having sunburn. It could be a monthly menstruation that activates it. Any kind of immunosuppressant, whether you're on drugs or maybe have not like a drug like prednisone, or maybe you've got another infection, chemotherapy can reactivate that. Just again, just having another infection, another fever. My husband hadn't had a cold sore in 30 years. And then last year, what made him get a cold sore was getting his second COVID shot. So, stinking vaccine. He was really sick for a day or so and got a cold sore. So any kind of stress like that on your body. The good news is usually symptoms are not as severe when they're reactivated. So we do produce a little bit of antibody to kind of dampen down the effects of that just a little bit. So oftentimes over time, it's not as bad over time. What can we do to treat it? Well, we don't have a lot. The drug that is most commonly used is called acyclovir, and there are several different kind of variations of acyclovir, but it's a nucleoside analog. So remember, we talked about this back when we were talking about mutagenesis in our genetic chapter 6. Where remember a nucleoside, remember what a nucleoside is? It's when you have one of the bases, guanosine, cytosine, whatever, along with the sugar deoxyribose. So that's a nucleoside. So if you have a drug that's a nucleoside analog, it's similar in structure to a nucleoside. So acyclovir, here's the structure. It's got guanine, which is the correct structure, but then not really deoxyribose, not really anything close to that, but still it's close enough to looking like the guanosine nucleoside that viruses don't usually have any good error checking, and they will incorporate anything that looks like a nucleoside into their DNA. But then of course, this is going to terminate the chain. There's no 3' hydroxyl group. Okay, so there's nothing for the next nucleoside to attach to. Okay, so that's going to halt replication of that viral DNA. Okay, so the acyclovir, this looks like it's injectable only, but it can also be given in pill form. It can be given as a cream, and it's supposed to reduce the length of the outbreak and maybe make outbreaks less common as well. For cold sores, there's not a lot, just kind of over-the-counter stuff like camphor, menthol, just to reduce that irritation a little bit. Okay, we don't have a vaccine. No vaccine. I've read about various herpes vaccines being at kind of various spots in testing. They kind of seem to be they languish in some clinical trial or another, and there's not any that's approved by the FDA. But they were working on one that's be given after somebody gets herpes, not to keep you from getting herpes in the first place. And it's supposed to not cure you, but again, cut down on the severity and length of any outbreaks. Okay, but that's nothing approved yet. So HHV3, called the varicella zoster virus, like I mentioned, this is the causative agent of chickenpox. Okay, and another one, again, that we're just passing around to humans. We didn't get it from chickens. It is named that because somebody thought the resemblance of the rash looked like the skin of a plucked chicken, apparently. Okay, how do we get it? Respiratory droplets. So you have to be in really, really close contact with somebody, closer than a meter. So things like coughing and sneezing, usually. So really close direct contact. And the really interesting thing about transmission with chickenpox is that people are infectious when they are in the incubation stage. Okay, do people have symptoms when they're in the incubation stage? No. All right, so that was kind of the tricky thing about COVID. People could be infectious when they weren't showing any symptoms. So that means that they're up around, you know, doing things that they normally do, getting people sick. Usually only a couple days for chickenpox. Potentially that incubation period is a little bit longer than that. So they're infectious for a couple days before they start, before the rash shows up, and then until the rash scabs over. So that can be, you know, maybe the rash might not scab over for 4 or 5 days, and then a couple days before that. Okay. And I thought this was a good time to just talk about something called R0. Okay, so we pronounce this R0. And this stands for the reproductive rate, or the basic reproductive number. Okay, so getting a little touch of epidemiology here. We use this, we can kind of mathematically figure out the basic reproductive number for a virus. So the idea is that that describes approximately how many people that one sick individual will infect. Okay, assuming that the entire population is vulnerable to the illness. All right, so this gets tricky when you have, oh, some people are vaccinated or some people have had it before. So it gets tricky to calculate it. But we typically calculate R0 for chickenpox. This is one of the most transmissible diseases. So that's typically calculated to be about 10. Okay, so if we look at the R0 for chickenpox, about 10. So meaning one person with chickenpox, assuming everybody was vulnerable, would get 10 other people sick with chickenpox. Okay, so that was why, you know, back in the days before the vaccine, almost every kid came down with chickenpox because it was so transmissible. So even more than that, with the R0 of 18, measles. Okay, that's one of the most transmissible things known. That's why when our vaccination rate dips a little bit, like it did a few years before COVID, we started to have little outbreaks, certain, you know, Disney World or whatever, because again, if people are not vaccinated, okay, they could very easily get it. Okay, so I thought that was interesting because of course there's been a lot of talk about this with COVID. If you compare flu, usually somewhere between 1 and 2 is what we consider influenza. So as long as the R0 is higher than 1, then it's going to keep going in the population. The original COVID was somewhere between 2 and 3. So we call now the alpha variant. Okay. So just a little bit more infectious than flu from the data that we have. And now the numbers get a little murkier because I think, of course, our data is, you know, some people are vaccinated, some people are not. But that Delta was somewhere between maybe somewhere between 3 and 5 for the Delta variant of COVID. And then I was trying to look up, well, what about Omicron? Some sources say it's 3 times as infectious as Delta. So that would put it at like 15. Other things say it's more like 7. So Omicron, you know, somewhere maybe about 7, a little bit more infectious, but again, estimates go a lot higher than that. So just sort of interesting to think about, because it does kind of make you realize, oh, I do have some personal responsibility. If I get sick, I'm likely to give it to, you know, X number of people. And of course, not everybody, hopefully, you know, people have had it, people have been vaccinated, but interesting to think about. Okay, so a little bit more about chickenpox. That was a little bit of a tangent, but I thought kind of an interesting one. What do the symptoms look like? Probably a lot of you have not had chickenpox, or maybe not seen anybody with chickenpox. Starts out, usually in kids, the first thing that you notice is the rash on the trunk. Sometimes when adults get it, they feel lousy for a day or two before the rash shows up. But it starts out on the trunk and then moves out everywhere. When I had it, I had a rash in my mouth and on my throat. I just remember being absolutely, really feeling lousy. I was 8. And just kind of other things, normal kind of viral infections, fever, malaise, loss of appetite. Okay, that rash eventually will scab over and be quite itchy. Eventually will be itchy. But usually it clears up on its own. And because we get good memory cells developed from that, usually people never get it again. So usually we only get it once. Now there are some exceptions to that. Sometimes when young babies get it, they maybe don't get as good memory established. They might get it again. Maybe somebody had a really mild case and they end up getting it again, but usually they get it once. Yes, question. What's that? So we'll get to that. Don't steal my thunder. We're going to talk about adults can get chickenpox. So we're going to talk about that. But if adults had chickenpox as a child, right, it can reactivate into something a little bit different, right? So usually though, when kids get it, they heal up and they're fine. So the question that people asked when the vaccine came out in 1995 is, why do we need a vaccine for something that is not a problem, like most people feel fine from? Okay, so 3 reasons why it's good to be vaccinated. Number 1, when you get chickenpox as a child, there is a possibility that you could get some kind of staph infection, especially you're scratching at that rash, you know, you tear your scabs off before they're ready to come off, you know, you can get a skin infection, which probably is not going to be serious, but that's a potential complication. More rarely, that chickenpox viral infection could turn into pneumonia or encephalitis, inflammation of the brain, but that was really, really rare. An even stronger reason to vaccinate kids, though, is because, of course, like HHV1 and 2, HHV3 can go latent. And it could go latently either the trigeminal, the brachial, or the sacral nerve. But when it does reactivate, about 1/3 of the people with chickenpox, it will reactivate. And when it does, it reactivates as shingles. Oops. Let's go. Okay, and shingles is a extremely painful rash. So it's not itchy, it hurts. And it usually will manifest most commonly kind of on the trunk region. So that term zoster, varicella zoster, zoster is Greek for belt. So it manifests either on one side of the trunk or the other. Interestingly enough, it will only reactivate in, one nerve's, one side of the body or the other. Here's where it's reactivated in the trigeminal nerve. And again, just very interesting, just one side of the nerve. But it, it can, and this painful rash, it usually lasts for about 2 weeks. But in about 10% of the people that have it, it will last longer than that. So 10% will have what they call post-herpetic neuralgia, where the pain of that rash just lasts for months and months and months. Okay, so there's the potential for this to be really, really quite severe and make somebody pretty miserable. And if an older, and it's usually as we age, we are more likely to have the chickenpox reactivate. But somebody with shingles, if they're in contact with somebody that never had chickenpox or was never vaccinated for chickenpox, can give that individual chickenpox. So there's a picture in your book of a grandpa with shingles holding his newborn twin grandsons. Okay, that's really dangerous. You don't want to give chickenpox to young newborn babies, right? So you would have to kind of, try to keep away from people. All right, and so the idea is if you were vaccinated against chickenpox when you were young, there's nothing to reactivate, right? You should never get shingles when you're older. Okay, so that's a good reason to vaccinate. And lastly, if you get chickenpox when you're older, you have a much higher chance of having complications like the pneumonia and encephalitis. So contracting chickenpox as an adolescent or adult, just much higher chances of it being a severe, very severe infection, having those complications. So of severe disease. Okay, I think some of you may have Dr. Baxter for lab. I think he got chickenpox as a teenager, and he had a pretty bad case of pneumonia along with that. So, either one, you want to get it when you're young. Ideally, get vaccinated as you're young. If you think you never had chickenpox and you've never been vaccinated, you can still be vaccinated for chickenpox as an adult. Might as well finish up talking about that. The vaccine that we have for chickenpox is called Viravax, and it's an attenuated vaccine. So remember, that's a live, weakened form of the virus. Usually it's given to kids when they're about 18 months, and then a booster about, maybe the time they go to kindergarten at about 5 years. I don't know if I would have to check to see if as an adult you would get the exact same vaccine. But you definitely can be vaccinated as an adult. We have a really good vaccine now for shingles. So people that had chickenpox when they were young, it's recommended for adults 50 and over. Okay, we used to have this Zostavax vaccine that was only maybe cut down your chances of it reactivating as shingles by about 50%. But now we have Shingrix that's greater than 90% effective. And if you do happen to get shingles anyway, then it's going to be pretty mild form of shingles. If you do get shingles once, you can get it again. Usually only not more than a couple times though. So it is possible to reactivate. This is just an inactivated virus. You have to get that, vaccine. You have to get it as 2 doses. When I had it last summer, that knocked me back worse than the COVID vaccine. So I was really, like, my whole body hurt after I had that second dose. But for me, you know, when I look at the risks, I don't want to ever get shingles. So I was, you know, glad to endure that for a day and not have to endure shingles. So really the only other thing we have to help with the itching of chickenpox or the pain of shingles is acyclovir. There's really not any other treatment that I'm aware of there. Do you have questions about chickenpox or comments or shingles? Okay, so that's good. I think we're in good shape to wrap everything up on Wednesday. Enjoy your day. We'll see you there then for our last lecture.